Hello and welcome. I'm Saeed from StoryPlanet.net. Dive right into the essence of the most captivating books without reading them cover to cover. Whether you're on the go, at the gym, or just relaxing at home, we offer you a unique and enriching listening experience. Today, we are exploring the book Life at the Speed of Light, a creation by J. Craig Venter. Life at the Speed of Light, 2013, details the author and his team's innovative creation of the world's initial synthetic life form. Delve into the excitement of their discoveries as they synthesize the first genome and study the teleportation of living organisms. Before we delve into these revelations, it's interesting to note that renowned biochemist and geneticist Craig Venter, known for his autobiography, A Life Decoded, has been featured twice on the prestigious Time 100 list, which recognizes the world's most influential individuals selected by Time magazine editors. With 10 key ideas to unveil, brace yourself for a deep dive into this captivating book on StoryPlanet.net. To start, prepare for a future where artificially created life forms will become prevalent. The text discusses a new world of genetic engineering, exploring a scientific team's journey to create artificial life by decoding DNA. It explains the possibility of leaving personal notes on DNA, synthesizing viruses, and potentially teleporting medicine to Mars in the future. Key idea number 1. Modern biology is rooted in a revised comprehension of life and its mechanisms. Physicist Erwin Schrödinger asked the fundamental question, "What is life?" in his lectures and book, What is Life? In 1944, sparking a revolution in science. His ideas inspired Watson and Crick's groundbreaking discovery of the DNA double helix in 1953, leading to a deeper understanding of genetics and the transmission of genetic information. This work laid the foundation for modern genetics research, emphasizing the importance of DNA as the carrier of genetic information. Key idea number 2. Artificial creation of life raises practical and ethical concerns. Since the 19th century, the scientific community has debated the concept of artificial life, sparked by Friedrich Wimpfler's synthesis of urea, challenging the distinction between organic and inorganic materials. This sparked debates between those viewing life as only chemical processes versus those who believe in vitalism. The current concern has shifted to whether we should create artificial life due to fears of moral consequences and loss of control. Despite popular fears depicted in literature and movies, the fields of chemistry, biology, and computing are collaborating to advance genomics and genetic science. Key idea number 3, discovery of DNA manipulation has revolutionized genetics. Scientists use restriction enzymes to cut and paste DNA segments for gene splicing, discovered in bacteria in the 1960s. This process allows for the insertion of genetic code into foreign DNA. Gene splicing has advanced significantly, starting with simple viruses in the 1970s and progressing to more complex organisms like bacteria and mammals. This technique has enhanced our understanding of genetics and biological processes, including the role of RNA in gene replication. Gene splicing has also contributed to the study of genetic defects and hereditary diseases like cystic fibrosis. Key idea number 4. Computer technology has sped up DNA analysis, helping in the quest to uncover the fundamental genetic code of life. Advances in computer technology and automated DNA sequencing machines revolutionized genomics in the 1990s, making gene sequencing faster and more efficient. This led to the founding of the Institute for Genomic Research and the first complete sequencing of a living organism's DNA. By comparing genomes, scientists identified common genes essential to life, leading to the creation of a synthetic chromosome containing essential genes. Ongoing research aims to synthesize DNA and further understand the essential components of life. Key idea number 5. Key point. A crucial experiment resulted in the creation of chemical DNA, enabling the development of synthetic life. The team synthesized a complete chromosome of a virus called PHIX-174 using computer code and chemical DNA. Phi-X174, a simple virus with 11 genes, was selected due to its well-known genetic structure. By using computer code to produce genetic information and assembling DNA building blocks in the right order, the team created synthetic DNA that successfully infected bacteria, proving that biological life could be produced chemically from computer code. Key idea number 6. In 2007, the authors' team created the initial synthetic genome of a living organism. The team synthesized a virus using synthetic DNA, then set out to synthesize the genome of a living bacterium, Mycoplasma genitalium. The scientists divided and synthesized the DNA into 101 segments, ensured accuracy through overlapping sequences, and marked their work with watermarks. They introduced the synthetic genome into yeast cells successfully, creating a living organism from a synthetic genome. The next big challenge is transplanting this synthetic genome into a cell to create a fully living synthetic organism. Key idea number 7. The team is known for successfully transplanting the first synthetic genome into a different cell. Scientists wanted to prove viability of synthetic DNA by transplanting it into a cell to become the celler's new genetic information, essentially transforming one species into another. They abandoned a slow-replicating bacterium for a faster one. Polyethylene glycol was utilized to enhance cell membrane permeability and protect DNA during transplantation. Discovery of a suitable host bacterium paved the way for the experiment. Key idea number 8. The first organism with a computer parent was created by transplanting synthetic DNA into a cell. The team used the polyethylene glycol method for transplantation tests on Max Mycoides' genome. A one-letter deletion in DNA sequencing caused initial failure, but the error was corrected, leading to successful transplantation. The DNA was activated, generating living, self-replicating cells controlled by synthetic genome. The team coined the new cell M. Mycoides JCVI Syn 1.00. This breakthrough marked the creation of synthetic life, but raised questions about reactions and impacts of artificial life. Key idea number 9. The public had mixed reactions to the first synthetic organism, with some people cheering and others jeering. The author aimed to demonstrate that DNA is the basis of life by creating synthetic life, challenging vitalism. Responses varied from considering it a significant moment to questioning the actual creation of synthetic life. Concerns included misuse of the technology for bioterrorism, highlighting the need for regulations. Balancing potential threats with benefits of biotechnology is crucial. Advancements offer increased access to technology for students and researchers. Future possibilities include digitizing and teleporting DNA for further advancements. Key idea number 10. Teleportation of DNA code between planets is being developed beyond science fiction like Star Trek. The concept of biological teleportation involves sending genetic information as electromagnetic waves for constructing life forms. This technology could potentially allow for quick medical solutions in space, such as teleporting DNA for antibiotic creation. It also offers opportunities for analyzing alien life forms without risking contamination or specimen damage during transportation. The application of this technology may lead to rapid diagnosis of diseases and designing medications, as well as enhance the understanding of life on other planets. In conclusion, modern biology focuses on understanding life through DNA, with advancements allowing manipulation and digitization of genetic code. This has brought about futuristic possibilities such as online DNA transmission and teleporting genetic code. Further reading recommendation is A Life Decoded by J. Craig Venter, detailing his role in decoding the human genetic code and his personal experiences in scientific research. Feedback on content can be sent via email to remember@storyplanet.net. Thank you for listening to this summary. If you enjoyed this exploration, we invite you to discover other fascinating books on storyplanet.net. Don't wait any longer. A multitude of books, stories, and knowledge await you there. See you soon on storyplanet.net.